Hi everyone, it's Christina, and this is by 233 Human Anatomy Physiology Lab, and today we're going to be talking about exercise 40 and 41, Anatomy of the Urinary System. So Professor Patterson has made a really nice set of online instructions for you, so I just want to point out a couple of things. So the first thing you have access to is a video of a pig kidney dissection, and so that's this link right here. And then, which is really short and informative, just kind of shows you the internal structures of the kidney in a macro view. Then there is a three-part video that this link right here is the first of 3 parts, and it kind of will go over the more macro structures of the kidney. And then just kind of going down through your lab, you're gonna go to a glomerulus, and so this link literally takes you to a beautiful magnified picture on histologyguide.com of a glomerulus. You can take a look at that. And then the next slide has a picture of the bladder, and you'll see both stretched and stretched meaning relaxed, and contracted meaning bumpy and thicker. The epithelium will be thicker on the contracted side and thinner on the relaxed side. Then you have a histology guide that shows you the ureter. I'll briefly go over these in my video, but since you have links and the histology guide is so useful with the things you can click on, I'm not going to spend a lot of time doing that. And then the second part of the 2-part model from the link above goes over the structure of the nephron, and so I'll spend a little bit of time on that. The one thing that the model video does a little bit differently than we do is the blood vessels names, and so I'll spend a little bit of time on those in slides. And then you're going to go on and do some stuff with urinalysis. It's pretty straightforward. Just use your book or textbook or lecture. All right, so this is what you are seeing when you look at the cut open sagittal section of the kidney. And I just want to highlight a few structures. So the fibrous capsule is this kind of outer layer wrapping around the kidney, holds everything in place, gives structural integrity. The cortex is this kind of outer area. So all this stuff is the cortex. All this stuff. The light-colored outermost area. This is where all of your nephron structures, your glomeruli, are going to live. The medulla is all of this middle stuff with the pyramids in it. Middle medulla. Renal columns are the areas kind of in between the pyramids. These are renal columns. So right here, blood vessels are going to go there. Renal pyramids are the little triangular shaped things, structures in the medulla. So this structure right here, this structure right here, those are renal pyramids. And then the renal papilla is just the tiny end, the apex of the triangle of the pyramids. So renal pyramid or renal papilla would be the last place where liquid would be leaving this tissue on its way out to the bladder. So then you have kind of all the areas where liquid, filtrate, and urine are going to go. It's filtrate when it's in this tissue. It's filtrate. Once it gets out here into these spaces, it's urine. It's done being processed. So a minor calyx is this first little cup where liquid is going to drip out of the papilla and go into this little air cup. That is a minor calyx. A major calyx is where 2 or more minor calyces come together. So this area right there, that's a major calyx. Renal pelvis is kind of the final gathering spot for urine before it exits through the ureter, and ureter is the tube going out, that tube right there. And so here's the blood flow slide I'm going to spend a little bit more time on. So blood comes in through our renal artery right there, and then the renal artery is going to go into a segmental artery, and this is the one thing that's not paired with all the others. So we can see interlobar has a vein matching it, arcuate has a vein matching it, etc. So segmental is the only one that doesn't have that, and it's this one right here that's going to go towards other areas of the kidney, this little artery right there. That's segmental artery. Interlobar arteries, for our purposes, now I want you to, they mention interlobular, and I want you to just listen to me. Interlobar goes between the pyramids. Interlobar, in between the lobes. Arcuate arteries are going to arc over the base or the wide end of the pyramid. Arcuate are going to go right there. And then cortical radiate are all of these arteries that are radiating out in the cortex. And then the veins are going to follow the similar pattern, where the blue ones out here are going to be cortical radiate veins. They're going to go into interlobar veins. Oh, pardon me, into arcuate veins first. Let me take that. So cortical radiate to arcuate to interlobar, and then directly into the renal vein exiting. There's no segmental vein at all. So this slide is all about kind of the nephron structures. And so I have a lot of vocabulary here. You can pause this and take a look. I'm just going to give you a really short tour. So our afferent arteriole is the thing that brings blood into the kidney, or into the glomerulus, pardon me. And so that's this one right there. Afferent, it's usually bigger. Then it's going to go into this glomerular space. This little bubble of stuff right there is the glomerulus, and plasma is going to ooze out between the pores of the capillaries and go into this capsular space right there. And then that arterial blood is going to exit again. So it's not venous blood yet. It's still an artery. So it's still a high pressure system. This one also is a high pressure system. And then it's going to go into the peritubular capillaries, which are all these squiggly veins around the tubes, peritubular capillaries. This is another flavor of peritubular capillaries. It's called vasorecta. You don't have that. You don't need to know that. Just know that capillaries around the tubes are peritubular capillaries. And so the blood's going to go in there, and it's going to go in there to reabsorb things that we want that might have oozed out in the plasma that we didn't want to get rid of. So after the glomerulus, you get the capsule space. We talked about that. Then the first tube is proximal convoluted tubule, where we're going to reabsorb interesting and useful things that we need. Then it's going to go down the loop of Henle and concentrate the urine more. And then it's going to come back up. So it kind of goes down the loop, and then it comes back up into the distal convoluted tubules, which are just another set of tubules. And then from there, it will go into this big collecting duct. And by the time we're there, we're doing very little modification of urine anymore. We're on our way to the calyx. And so this is just a closer view of the nephron itself and of the glomerular structure. And so here we have our afferent arteriole bringing blood in. Here is our capillary bed, where our red blood cells stay inside, but the yellow plasma oozes out into this capsular space, and then it will gather and go into the proximal convoluted tubule. So the blood kind of comes in, presses out, squeezes out the plasma, and then it exits through the efferent arteriole. This is the afferent arteriole, arriving, exiting, and then this blood is on its way to the peritubular capillaries. On the side of this page, we can see histologically what you're going to be looking for. So this thing right here is this thing. So this is the glomerulus, and then this is the capsule space. And then here we have a smaller version of that, And then all of these little structures out here are proximal and distal convoluted tubules. All of that stuff is proximal distal convoluted tubules. And then sometimes we can see some collecting ducts also in between. And all of this is happening in the cortex. If you see a glomerulus, you're in the cortex. There are no glomeruli in the medulla at all. And when you click on the link for histologyguide.com, this is what you see. So we can see the glomerulus right there, and then here's the capsule space, and then we might see some arterioles entering. It's hard to tell, but all you have to know is that this is a glomerulus, and that all of these structures out here are tubules. Tubules, tubules. All right, so when you click on the bladder slide, I know not everything is in view, but I wanted to be able to show you the menu of stuff. And so on this side, you're going to see kind of a thicker epithelium. So this side is going to be contracted. And this is empty, an empty bladder, right? And then on this side, the epithelium is thinner. And so this is stretched, relaxed, and full. And so just kind of zoom in. You can see you have nice little links to click on the epithelium in both places, just kind of be able to tell contracted from relaxed. And then here you're zoomed in on a ureter, and so basically you're just going to have to be able to identify this slide, this structure on the slide. It also is going to have transitional epithelium in it, and so you're going to want to zoom in on the epithelium and take a look at what those cells look like. And that is pretty much my review of the urinary system. I'll see you next week.